Hello and welcome to another episode of the Omics cast. And in this episode, we're going to talk about the microbiome and the immune system. My name is Alberto Vilella. For those of us that still don't follow me on Twitter, you can follow me on twitter.com/Alberto Vilella, all one word. And you can also contact me on LinkedIn if you want. I'm happy to answer any questions that you have. And this slide deck is available upon request. And in this episode, we're going to talk about the recent preprint in bioRxiv, the analysis of a paper on the gut microbiome and the effect it's got on immune cells. It's a paper with where the corresponding author is affiliated to the Memorial Sloan Kettering Cancer Center, and we're going to discuss what this paper, the results of this paper, what they are and what are the implications in this field. So in the abstract, they basically say that they wanted to understand more about how the gut microbiota influences the immune system. And they did this by studying very closely a group of individuals that had bone marrow transplantation. And they were recovering from a chemotherapy-induced immune ablation. So it's if you want, an extreme case of altering the immune system and then seeing, following up this patient and seeing how their microbiome reacts to that large alteration. And then what they see is that this aggressive treatment causes large shifts in both white blood cell and microbiota populations. And so what the results are is that they have identified different drug treatments and associated it with clinical metadata that show associations between microbiota and blood cell dynamics, white blood cell dynamics. And they say, for example, Streptococcus are associated with increases in lymphocytes. Actinomycete are associated with suppression of lymphocytes, and then some other ones here that I'm not going to even try to pronounce are associated to increases or decreases in the population of white blood cells, monocytes, neutrophils, etc. And then this is, if you want, a very comprehensive analysis to see after the ablation of the immune system through chemotherapy, bone marrow transplantation, what happens to the microbiota and how they can see the link between the two. So the most striking figure in the paper is this one, where they show the different time lapses of what these different patients, what happened to them. And then we can see here two very distinct examples. Patient one in the middle, that it's got population of neutrophils here in blue going up after the ablation and staying sort of at a medium to low level. The lymphocytes going up and then decreasing slightly, and the monocytes going up and then decreasing periodically over time. And then here's the plot where you can see in different colors the we can call it an ebb and flow plot of different populations of bacteria in different colors, and the legend is the bottom left. So an example of what happened to one patient. And then strikingly next to it is patient number 2. And look how strikingly different these populations of white blood cells are. Here we see a massive increase in neutrophils, and then a different pattern altogether from patient 1 from lymphocytes and monocytes. And they coincide or correspond to also markedly different populations of different strains of bacteria in the gut microbiota. So at the bottom here, we can see some associated clinical treatments, different drugs or different other treatments or conditions identified. And all of this look together is the strongest link is between the white blood cell populations and the microbiome of each individual. A little bit about the bioinformatics analysis, not to go too deep into it because a lot of it is known already in the field. It's been used for ages. In some cases, stool samples collected from patients and sequenced using primers for the 16S gene. It's a very conserved part of our genome. And so it's a way of finding of our genome and genome of all sorts of species, finding sequences that can classify what organism they belong to. Clustering with UPARs and then comparing against RefSeq RNA, and then diversity calculated by the inverse Simpson index here. There's more details in the methods. If you want to look at it at the paper, I thought this was enough detail for what they said. So essentially, lots of sequencing, 16S, clustering, analysis of diversity, and finding out the relative proportions of the different species in the gut microbiota. So the authors say that basically there's this systemic homeostatic link that could help understand the modulating role of a healthy microbiota during immunotherapy, which, and very importantly, holds potential for microbiota therapies, which is something that we're starting to see in the field, and it's showing very promising. So as a summary, what happens in our gut, what cells, what individual bacteria, species, etc., live in our gut, has an influence in our health, as you can see in this picture. And this interaction is by a different biological molecules. Some of them are listed in this picture, which is from a review of the microbiome studies that have been taking place recently. And this can be altered in different conditions. For example, here we see a normal individual where some beneficial bacteria or pathobions are kept in check, and their relationship with the lining of our stomach and intestines is kept in check, and the T cells and other white blood cells react and compensate for keeping in check these pathobions, etc. In diseases, and the one displayed here is IBD, inflammatory bowel disease, then the gut microbiome, the proportions are altered, and they call this dysbiosis. It could be related to genetic factors, but also environmental factors like diet, stress, infections, treatments with antibiotics, for example. And then this dysbiosis has a contribution to what happens to us inside on the bottom part of this plot, inside our bodies. In this case, chronic inflammation due to IBD. A lot of this is the power of NGS, next-generation sequencing. We couldn't have done this analysis with this high level of precision before the advent of next-generation sequencing. And this is mostly due to a lot of this diversity, finding the diversity of the samples, requiring lots of sampling, deep sampling of what we have inside us in the microbiome and the gut microbiota. And this is because many of these follow a power law distribution or long tail distribution. Power law is a type of long tail distribution where sometimes the pathogenic or really interesting ones are in the low popularity end of this tail. But if you're only sequencing certain amounts, you're either going to find what you're looking for, or you're only going to find a small slice on the left-hand side here. So the power of next-generation sequencing here is crucial to be able to inform the microbiome. As a conclusion from this quick review that I did on this paper, in bioRxiv, microbiota therapies could help in recovery from immunosuppression in clinical setting, including cancer treatment. So very important for people. Not only is it important that we diagnose cancer early, but also that we treat it early and successfully, and then we keep up, and that we can deal with all the secondary effects of the treatments, including the recovery of immunosuppression in our gut microbiota. The balance of microbiome relative diversity can be influenced by external effects, and this has a knock-on effect internally on us. And here we've seen it in this paper in the immune system. And NGS is opening up new avenues of discovery due to the high resolution signal that we managed to get from sequencing so cheaply compared to what we were able to do 10, 20 years ago, the samples that we're interested in. Thanks for listening. As a reminder, you can follow me on Twitter, twitter.com/albertvilella. And I'm happy also that you contact me through LinkedIn, and I'll answer any questions that you have. And this slide deck is available upon request. And I hope to see you soon in the next episode of the Omics cast.